Discussion today. It's going to be focused on the cell cycle, both how the cell cycle functions normally, as well as how the cell cycle can begin to function abnormally and what problems it can cause. So our essential question that we are seeking to answer by the end of this video is, what are the steps and purposes of the cell cycle? So basically, the purpose of the cell cycle is anytime that a cell needs to, or an organism needs to grow or repair itself or replace old broken down cells. And it's also used as a means of reproduction in single-celled organisms. So the results of the cell cycle at the most basic level are going to be to duplicate accurately the DNA that's in the chromosomes in order to create two identical daughter cells. So we're going to very quickly look at the cell cycle in all of its depth, and then we'll start applying it and making models of it. So the longest part of the cell cycle is going to be interphase. And interphase is actually broken into three subphases that vary in length and of course in purpose. And so because cells are going to be limited by their surface area to volume ratios, the ratio, remember, has to remain small in order for the cell to transport materials and for the DNA to be effective. Interphase then becomes the longest portion of the cell cycle. And again, it's broken into those three subphases. So the first phase is going to be G1. And if the cell cycle were in a 24-hour period, the cell would spend 12 hours in the G1 phase. The cell goes about its everyday functions. It continues to grow. Once it starts to get too big, though, to function efficiently, extra organelles begin to become produced and the volume starts to purposefully increase. So again, this is the longest phase. After G1, so the cell has gotten to the size where it knows that it's going to have to start to divide in order to remain functional, the cell goes into the S phase, which again in our 24-hour period would be approximately 7 hours. All that happens in the S phase, and I say that with levity, is that the cell carefully duplicates its DNA. Finally, we get into the shortest phase of interphase, and that is going to be G2. Notice that this is only going to be approximately 4 hours. This is the final push before the cell goes into its division phases of the cell cycle. So final duplication of the organelles is completed, cell volume is increased to its max limits, and then after final preparations are finished, the cell is going to enter mitosis. And remember, this is the shortest portion of interphase. And so looking at the cell cycle as if in a modeling format, you can see that we have G1S, G2, interphase is all of that, and that is the cell cycle overall. But we can just kind of do the very quick, most important events that are happening in each of these phases. So in G1, or first gap, extra organelles are produced, cell increases in volume. In the S, synthesis, the cell duplicates its DNA. In second gap, or G2, organelles are finished being produced and the final volume is increased. So once a cell reaches that portion of G2, it moves into the final phase of the cell cycle, which is mitosis. And mitosis is also broken into subphases, and we're going to learn much more about these subphases later. Just know that mitosis is the M phase. It's where the cell is actually going to begin to pull apart, and then mitosis is going to be followed by cytokinesis, where your two daughter cells are actually produced. So in prophase is the first phase of mitosis, that's preparing to divide. Metaphase is the second, and that is where the chromosomes align in the middle. Anaphase is where the chromosomes pull apart. And telophase or telophase, either is okay. Two nuclei are formed. And then, of course, we have cytokinesis, which is outside of the M phase, but that's the final point where we have two actual daughter cells produced. So if we go back to our cell cycle model that we had before, you can see now we've entered in the interphase information. to go along with our interphase information. And so now we add in cytokinesis, and there's a model of the cell cycle. So there's this nifty little acronym to help you remember the cell cycle and its order, and it's IPMAT plus C. So we go interphase, prophase, metaphase, anaphase, telophase, and then cytokinesis. So hopefully that can help you to recall what you need. So that's if everything goes normal with the cell cycle. But unfortunately, as life has taught us, things don't always go as they are meant to go. So what controls the cell cycle? Well, some cells divide frequently, like skin cells, and others only when they're wounded, like your liver cells. And some cells, like your muscle and nerve cells, never go through cell division, which interestingly enough, when they need to divide, they do it through a whole other process. So there is a protein in the body called a cyclin, and cyclin is a protein that is going to be responsible for regulating the cell cycle. So once again, everything comes back to proteins in our body. There are different cyclins then between each phase of the cell cycle that act as checkpoints. And these checkpoints' job is to make sure things are progressing normally. When they do not progress normally, then the cell has to take action, and that's where things kind of get interesting. So there's a lot more cyclins than A, B, E, and D, but these are the ones that we're going to be thinking about in terms of our discussion of the cell cycle. So cyclin E is responsible for checking a cell between G1 and S phase and moving it forward if everything is okay. Cyclin A checks the cells between S and G2 to make sure everything's okay before they move on. And cyclin B checks the final preparations of G2 before it allows a cell to move into the M phase or into mitosis. So we talked about, kind of hinted around, that we're going to talk about what happens if things go wrong. So first and foremost, the checkpoints in the cell cycle are going to maintain the process so that all the copied cells are normal and properly functioning. Sometimes, though, the cell cycle, the process, is going to make mistakes. So what happens then? The process is going to stop at the checkpoint that identifies the issue, and the issue will be corrected through different various means. If the mistake cannot be corrected, the cell is going to enter into apoptosis, which is programmed cell death. So sometimes even the means that we have, or that the body has, to keep our cells copying correctly don't work. And so when the cell cycle goes completely wrong, you can end up with conditions like cancer. So sometimes the mistakes in the cell cycle and cell division cannot be corrected by the cyclins and those mechanisms, and somehow the cell avoids apoptosis. Then what happens? Well, cells generally stop growing when they come in contact with other cells, and they only grow rapidly in response to injury or growth. But sometimes they have an inappropriate need to divide, and they don't care if they come in contact with each other. So this is actually the condition of cancer, and cancer is nothing more than the uncontrolled cell division. And I say nothing more, even though it's significantly more in the effects that it has on the body. So these cells do this by not stopping growing. So when they're in contact with each other, they avoid apoptosis, and they also get through those cyclin-based checkpoints. In essence, these cells never go into interphase. They're always in mitosis. So they keep dividing, and then this uncontrolled division forms masses, which leads to tumors, and then it can also progress into other forms of cancer. So go ahead and summarize your notes on this topic by answering your essential question in the summary section. And then go ahead and look at your page 9 and answer the 2 check your learning questions.